When global policymakers gather at elite AI summits to discuss safety, they are focused on a very specific set of problems. They debate deepfakes, large language models, and text copyright. While they focus on text, a silent enclosure of the global biological commons is occurring without international oversight. To understand this, we have to look at the gatekeeper of artificial intelligence, the tokenizer. AI doesn't read words or DNA sequences as continuous ideas. It breaks information down into distinct mathematical units called tokens. The foundational tokenization algorithms built by major tech companies are optimized strictly for Western languages and Eurocentric text structures. They look for common syllables and word fragments to map data efficiently. Forcing biological data through an algorithm designed for human speech creates an architectural blind spot, one that compromises global scientific sovereignty. Biological sequences, like DNA from a climate-resilient crop or an indigenous medicinal plant, do not have syllables. Their language is dictated by reading frames and evolutionary structural motifs. When a standard text algorithm, like Byte-Pair encoding, attempts to read a genomic sequence, it treats the genetic base pairs like random English letters. It fragments a singular, functional gene into arbitrary, mismatched pieces. Because the algorithm is fighting against the natural structure of the data, it generates an excessive number of mathematical tokens just to represent a tiny snippet of DNA. This structural inefficiency is called token bloat. The AI is then forced to use immense computational power to guess where a gene starts and ends. A single misplaced token can change a predicted protein fold or metabolic pathway, introducing artificial hallucinations into the biological model. If a model misidentifies a drought-resistant trait or misses a new crop mutation, the result is a failed harvest. These hidden structural flaws turn computational guesswork into real-world food insecurity. Processing these inefficient, fragmented biological datasets requires massive, expensive supercomputers. This hardware requirement creates an artificial scarcity. It effectively prices regional and global South laboratories out of participating in advanced molecular research. The computational power remains locked within wealthy tech hubs, far removed from the local communities who have preserved and managed this biological diversity for generations. Regional researchers in sovereign states cannot protect a bioeconomy if they do not computationally control the underlying scientific architecture. Meanwhile, commercial AI companies use automated crawlers to harvest raw native plant profiles and crop traits from open-access scientific repositories. When these native biological sequences are pushed through opaque closed-source tokenizers, the process strips away their geographical map coordinates and sovereign origins. International treaties, like the Nagoya Protocol, were designed to regulate physical biological materials. They are currently unequipped to handle digital sequence information, or DSI. As a result, sovereign biological heritage is converted directly into privatized computational capital. The nations originating the data are left with zero economic returns. Without clear frameworks for digital provenance, the collaborative ideal of open-access global science becomes a vehicle for digital extraction. The scientific countermeasure relies on developing open-source, traceable molecular tokens. These custom algorithms read biochemical structures correctly and natively embed unalterable geographic origins directly into the data. Because these tokenizers are native to biology, they reduce the overall data size. They compress genetic processing pipelines and eliminate token bloat. This level of efficiency enables Frugal AI, allowing decentralized regional laboratories to run highly complex molecular simulations on standard, low-cost hardware. By natively optimizing data right at the point of sequencing, we can re-engineer global laboratory workflows. We shift the research process from slow, analog observation to instant, democratic precision science. To bridge the communication gap between the laboratory bench and the parliament floor, researchers can utilize science policy translation notes. This framework converts complex computational token metrics into clear, actionable legislative policies. Immediate regulatory goals include establishing sovereign genomic data vaults and legally mandating biological token provenance audits for any commercial AI tool operating within a nation's borders. Shifting away from text-based algorithms in favor of traceable genomic tokenization secures our biological data. It ensures that artificial intelligence protects global biodiversity. while guaranteeing equitable scientific advancement for all nations.